Hi there, my name is Paxton Barco. The date is March 7th, and I am going to walk you through in this video how to digitally label images. To get started, I'm just going to tell you, I'm just going to move the computer screen down. I hope that's okay, just so that you can better see my computer and everything, but you'll still hear my voice, and obviously it's still me right here. Okay, so getting started on digitally labeling um, images. So the first thing that I want to say is that the most important part about digitally labeling images is making sure that you can effectively do it. So making sure that you have the correct tool, basic editing software. So I personally like to use PowerPoint, which I have open right here, but you can use Microsoft Word, you can use Google Draw, you can use Pages, whatever you want. You just want to make sure that the tool you use is going to allow you to do a few different things. First off, you need to make sure you're going to be able to import the image and that the image will be a nice quality so that you can label it. You're going to want to be able to draw lines, which I'm going to be able to do right here. And you want to be able to add text boxes because that's how we're going to describe what we're labeling. So as long as you've ensured that the tool you use will allow you to do all these things, you are set to go. Now, the next thing that I want to say before we actually label our images is that we want to do our research. We want to understand what we are actually labeling. So one of the most important things that we can do, obviously, is rely on our textbook and also rely on our lab manual, which gives us a lot of great details, sometimes includes pictures that are very similar to the images that we will label, and we can learn a lot of important information so that when it comes time to label our image, we actually know what we're looking for and it's easy and efficient and it's correct. So I know that in this case, I have to label 3 different structures. I need to label an acidophile cell, a basophil cell, and a pituitary nucleus. So I'm going to consult my lab manual. This is the area about the pituitary gland, the area of the manual. And I'm going to look down here at all the photos that are also available to me right here so that I can understand really what I'm looking for. And once I've done that, then it's time to actually start labeling. So, first important step is importing your image. And this is very important. You want to make sure that the image that you import into your editing tool is a high enough quality that you're going to be able to distinguish the different structures that you need to label. And it's just going to make your life that much easier because you'll be able to zoom in and really get a good look. So I am going to close. Sorry, one second. I'm just going to close this lab manual. I have my PowerPoint open and I'm going to copy the image. You can also drag it, you can download it and then upload it, whatever you want. I'm just going to copy it, but then I'm going to make it much bigger in PowerPoint. And this is going to allow me to zoom in, etc. And as you can see, it's still maintaining enough of the image quality that I can still see what's going on. And I'm planning to screenshot this later, so it means that it's okay if the text, if the image box goes outside the PowerPoint slide, etc. Okay, so now I've made this to a pretty good size. I know what I'm looking for and I'm getting ready to label. So after examining the different images in my lab manual and I feel ready to label, the first thing that we want to distinguish is between the anterior and the posterior pituitary, because I know that the 3 structures I need to label are found in one of those 2 parts of the pituitary. So now this is pretty simple for us because we know that the anterior pituitary tissue is dark stained, so kind of this bottom half of the image, and the posterior pituitary is lighter stained. So that was pretty simple for us thanks to the information we had in our lab manual. So now we can move on to labeling an acidophile cell and a basophil cell in the anterior, so this lower portion of the pituitary. So acidophile cells are cells that have a pink cytoplasm. This is looking under a microscope at an image like this, and a dark nucleus. So I'm going to zoom in on my image. There we go. You can see me zooming in. I'm going to zoom in on my image, and what I'm going to do is just find an area as best I can that is pink. I want to see that pink cytoplasm. So a good significant amount of pink, so it's very clear that I am labeling an acidophile cell. And I think I'm going to choose right in this area because I see a good amount of pink and very faintly maybe a nuclei in there, so that is perfect. That's an acidophile cell. So I'm going to use my line tool and I am going to draw a line, and I'm going to draw the line, I'm going to put it right on top of the area of the cell that I want to point to, and then I'm going to draw the line outside of the entire image, and I'll explain why I do that in a minute. And I'm going to change it to black, which I will also explain why. Now we're going to add a text box right here next to it, and I'm going to type in acidophile cell, make sure it's the right, better size. Awesome. So as you can see, it's pretty straightforward. It's really easy once you know what you're looking for. Okay, so now we will label a basophil cell, and then I'll explain a few things about actually labeling. So the basophils appear as darker cells with a purple or kind of bluish cytoplasm. So again, I want to stay zoomed in, and I want to try to find an area that is clearly a basophil cell. So take another line, and I'm thinking a really good example is maybe up here. This is a good example of, it's very purple bluish, distinguished from the pink, and definitely a basophil cell. And again, I'm drawing my line from touching right on top of the part of the image, and then drawing outside the text box. And now I will label basophil cell. Again, make this a better size and pull that up. Awesome. So one quick thing I want to point out when you're actually drawing your lines, it doesn't really matter what color you use for your lines. However, I will say that I recommend using a color that contrasts really nicely with the image. So that's why I've chosen black. It stands out against the image, and it's very clear what I'm pointing to. And like I said before, I'm going to put the start of my line right on top of the cell or structure that I want to show, and then I'm going to pull it all the way outside of the actual image and add my text box there. And the reason that I do that is that way the text box or the line doesn't get hidden within the image. It's very clear. Everything just remains really clear. Okay, now finally we've labeled the structures of the anterior pituitary. So now I'm going to zoom back out. I'm going to go up a little bit. I'm going to zoom back in to the posterior pituitary portion. And we know that we need to label a pituocyte nucleus. And I know that I'm looking for basically a tiny kind of circlish, darker structure. This is pretty easy to see. I'm going to choose that one up near the top. That is pretty clearly a nucleus, a pituitite nucleus. Make that black again, and then we're just going to add in pituocyte nucleus. Okay, awesome. And then obviously double checking our spelling, etc. We will add that. Okay, great. And again, following the same protocol as the dark line, pulling it outside the box and then labeling there. So hopefully you can see that. Okay. All right, well, that's pretty much it. If we zoom out now, we can see all of our different areas that we've labeled. So really the last thing you have to do once you've labeled all your structures is just double check your work. Make sure that your lines are in contrasting colors. Make sure that your text boxes are spelled correctly, that they also stand out in a nice contrasting color. Make sure that your image hasn't moved, you're still pointing to the correct structures. And once you've done that, you are good to go. So thank you so much for listening. I hope it's been very clear of how to digitally label an image. And the last thing that I will say is when you're all ready to go, my preference is to screenshot it and then drop that into my lab manual. And that's my way of knowing that the image is going to be around the right size for a lab manual. So thank you so much, and I look forward to your feedback.